Thanks again to the National Academy for hosting us here. And I am hosting the session, Coccidioides, the Organism and Hosts, and it's an appropriate thing to be doing this afternoon because I was supposed to be in Tucson giving a fungal plant pathology lecture. And one of my opening slides in that lecture is the disease triangle, which talks about the association of the environment, a conducive host, a susceptible host, and an appropriate pathogen, all together working to cause disease. And in this session, we are going to have speakers talking about the environment, the host, and 2 talks on the pathogen. And we'll start with a talk by Bridget Barker from Northern Arizona University on ecology of the environment. We will have 2 virtual talks coming up in this session. Bridget.
Okay, so I was moderating this, so in theory I know how to work the clicker. So yeah, thank you so much to all the efforts that the Academy has put in. It's been a great meeting, and I'll talk to you very broadly about the ecology and evolution of the organism that causes the disease. So first of all, I want to make a land acknowledgement. This is very important for me specifically, as well as within the state of Arizona. When we think about the fact that 30% of the land in our state is owned and managed by various tribal units, and a lot of the work that we do and a lot of the sampling that we do occurs on indigenous lands. So I promised that I would talk about the 2 species. And so there are 2 species, Coccidioides immitis and Coccidioides posidossii, and I'll talk about the distribution of those and the biogeography of these particular organisms. But I wanted to first point out that we do know that there are significant phenotypic differences between the 2 species. We have only tested a very small number of phenotypes actually for this organism. It's an area where we really need to improve our understanding. So this is work that I actually started. So this is some of the very first work that I did on Coccidioides 20 years ago. Just frightening. And it was under the tutelage of Dr. Orbach and Dr. Galgiani. And I remember the day, because we were trying to get the organism out of the soil, and we were trying to develop media and conditions that would allow us to get the organism out of the soil. And so one of the suggestions was grow it at different temperatures. And so I did, and I saw this really odd phenotype that some of the strains weren't really growing at 37 degrees. And John actually came into the lab and said, you know, these are the 2 different species, right? Maybe he doesn't even remember doing this. But it led to a really elegant study. So we were able to collect a lot of isolates across the entire geographic range and definitively show that Coccidioides immitis is inhibited when we grow it on a plate at 37 degrees. The reason, the mechanism is not known, but it is a significant phenotype. And I'm sure there are other phenotypes, such as pathogenesis potentially, or other ecological interactions that are important. So we see strong biogeographical patterns amongst clinical isolates, because that's mostly what we have, are clinical isolates and not environmental isolates. We find Coccidioides immitis, California and Baja, Mexico primarily, but then of course that disjunct distribution where we have this isolated population up in eastern Washington is also Coccidioides immitis. And then Coccidioides posidossii is throughout the rest of the range. So these colored pie charts just sort of illustrate the distribution of various alleles in those populations, and they're color-coded so that genotype is being matched to the location of these organisms. So that was with microsatellite markers, which is just a few targets within each genome. Now we've been able to do phylogenomics, so actually taking whole genomes and comparing all of the SNPs, all of those polymorphisms across the entire genome, and clearly from this, we can see, so if you're not used to looking at phylogenetic trees, the length of the branches mean how long those are separated evolutionarily. And so you can see pretty quickly that Posidacea has very, like we had to shorten it to make it fit on this image, right? So it has very long branch lengths. So these are separated by millions of years of evolutionary history. And then within each species, there's additional separation. So these are color-coded, just again, so you can see them very easily, where we have this Texas, Mexico, South America clade within Posidacea. We have a separate clade for Venezuela, Guatemala, which we have called the Caribbean clade. And then we have an Arizona clade. And then in blue here is all the Immitis isolates. So you can see a couple of different patterns. So some of these branches are really long when we look at some of these populations, and some of them are very short. So within the green box, I'll just have you focus on that for now. When we isolated genomes from Brazilian isolates, you can see they're kind of like all of those dots represent individual isolates, and they're all in a really straight line. That's telling us that that population seems to be particularly clonal. So they're all very closely related to each other. But if we look at the rest of that clade, so Texas, Mexico, South America, those branches are much longer. So that's telling us that there's more variation potentially within that grouping when we compare it to Brazil. So this is new finding. This is actually unpublished right now. And this is work from primarily Dr. Marcus Teixeira, who will be talking, I think, tomorrow. And we'll be giving some information about more of these types of patterns that we're starting to observe in South America. You can see the same pattern. So I've highlighted the group from Venezuela. So we see that same pattern in Venezuela. And then down in Washington, too. So in that blue grouping, you can see that little box area that shows Washington. And it shows that same sort of clonal population compared to the massive amount of phenotypic and genotypic diversity that we see, say, in Arizona. So this is an interesting pattern. What does it mean? Well, we don't really know yet. So there's a lot of work to do. This is a relatively recent finding, as I said. And so there are a few different possibilities when we think about observing this type of population structure. So we know that the population structure varies. Some regions harbor isolates that seem to be highly recombining, like Arizona. Another way that we can measure that is to try to understand if sexual recombination is happening. So fungi don't really have male and female, right? But we look at different mating types. And so for coccidioides, it's what's called MAT1-1 and MAT1-2. And you can sort of think about these as male and female, although, of course, that's not correct, but just for the simplicity of the argument. In the Arizona population, we find those alleles in a 50-50 ratio in the population. So that would indicate that that's a highly recombining population, sex is probably happening, and there's a lot of diversity. When we look at those isolates from Venezuela, from Brazil, and from Washington, we only see one of the mating types really present in those populations. So are these newly established populations? Is this sort of the evidence that we've been looking for to think about, okay, the organism is potentially moving, dispersing to these different areas? Are these new founder populations? That's possible. Are they maybe locally adapted? So maybe these also represent marginal habitat. So places cocci can live, but maybe it's not super happy. So it could also be that these are also highly locally adapted to survive in a particular marginal habitat. So these are both sort of formal hypotheses that we're now exploring. The importance of understanding genetic variation, I think I've shown this slide a few times, but I just like to kind of remind everyone, we can do a lot of genome sequencing, but why? Why are we doing that? Why are we trying to understand these populations? For me, it's interesting to think about how that genotypic information can result in phenotypic changes. So are there differences in pathogenicity? Do some of these strains cause more severe disease than others? Do they cause more severe disease in Brazil, or less severe disease in Brazil with these particular isolates? Is that why we don't see very many cases reported there, right? Is it a reporting issue, or are these actually maybe lower pathogenicity strains? Are there differences in treatment? I think this has been a common question that I get asked a lot. So, when we're trying to design a vaccine, we want to make sure that we're covering all of the genetic variability as well. Differences in antifungal disease or antifungal drug resistance, and that's emerging. So again, is that maybe one particular population or maybe one species versus the other is more or less antifungal resistant? And then for a lot of the stuff that we've already been thinking about in the epidemiology piece, are there different associations in the environment? So is this genetic differentiation reflecting different hosts, different natural hosts in the environment, different responses to heat, different responses to precipitation regimes? And does that result in different outbreak patterns? So when we look at differences between California and Arizona, are we also observing differences that is a direct reflection of the different species that are present in these areas? This was already discussed earlier, right? I just like putting it in the terms of, vegetarians versus carnivores. And so we think of cocci as and the group of organisms that contain coccidioides. So in red is outlined that whole order of organisms, the Inigonales, and we have an expansion of genes associated with breaking down things like keratin, bones, hair as a food source. But cocci is really unique in this aspect that it has both a reduction of plant-associated genes and a very large expansion of those metalloproteases and cetylases, et cetera, that are necessary for breaking down that complex keratin structure. I also think a lot about how I'm going to be linking genomics with ecology and how we do that. And I think Jason Steich is probably going to speak to this a little bit more specifically in his talk next. So thinking about gain and loss of genes, right? So just as I gave us a small example of that, that was based on a very small data set. We want to expand that to looking more broadly, both between the species, comparing to other genomes, now that we have a lot more genomic data. We want to look at genes that are potentially under positive selection. So genes that are rapidly evolving, maybe genes that are associated with antigens, right? So those might be a way that we can detect novel antigens in the genome by using those approaches. There's a lot of work, and I think Dr. Sill will talk about that in terms of looking at transcript expression and how does, what those genes are actually turning into in terms of mRNA and then protein, how does that inform us about how the organism is able to switch between those different life stages, right? So how many arthrochonidia can it make? Do each of those arthrochonidia disarticulate in a way that makes a single arthrochonidium that can get deep into the lungs versus long chains of arthrochonidia that kind of are sticking together more? And so does the disease outcome differ when you're exposed to those different structures, right? And so I think those are going to be very informative pieces of knowledge that will help us understand this organism and its disease progression. Right, so we'll get to the endozoan hypothesis. I think I have a few more minutes at least. So this is an environmentally acquired disease, yet we understand so little, fundamentally little, about where the organism grows, when is it making lots of arthrochonidia, when are those arthrochonidia getting into the air, if they're getting into the air, how far are they going, et cetera. And it's so critically important to understand that because we are infected directly from the environment. So I have spent a good portion of my career trying to understand this. So both looking at the organism, growing it in the lab, seeing how it responds to various stresses, but also thinking about ways to get out into, well, mostly excuses to get out of the lab and go into the desert. How do we develop techniques to actually detect the organism in the environment? So there's a lot of efforts to do this. A lot of the methods gave a lot of false negatives and false positives. The culture-based approach is really intensive or doesn't work. So we developed an assay using a real-time based TACMan probe-based assay to detect the organism directly from DNA extracted from soil. So if you're interested in this, I just kind of wanted to give a little shout out for our tool. Unfortunately, we found that maybe it doesn't work as well in Texas soils and South American soils. We're actually going back to the drawing board a little bit to tweak this assay a little bit so that it might work better in those situations, but stay tuned for that. So what do we know about the work that's been done? How can we try to understand what zones and what regions are important for coccidioides? So some of the biotic factors, right? So there are specific animal species in North and South America, but there's also other microbes. And I think the microbes get neglected in general. They don't just cause disease, right? They interact with each other in the environment. So we need to start looking at some of these other microbes because arid lands are characterized by a structure called biocrusts. Biocrusts are natural organisms that grow on the surface of the soil and may represent a way that we can actually do ecological restoration in some of these sites. So that's work that's ongoing. There's seasonal moisture patterns. Obviously, we've talked a lot about temperature, drought. Desert soils themselves are very specific, so they have fairly high pH. There's a lot of salts and minerals that can be at very high concentrations in these desert soils. So this is ongoing work. We're really trying to understand what specifically is associated with cocci in these environments. One thing that we know is associated for sure is based on work by Dan Coleth, who will be talking tomorrow. This is his master's work where he went out and did this survey. John already talked about this. And so burrows, those animal burrows, do represent a point source of infection and outbreaks, and for us, do represent a majority of our positive samples. That's not to say that we don't find it in other places, and that's not to say that we understand specifically that it's the animal hosts. It may be the burrow environment itself. And so those are things that are ongoing, and you heard some of that work today. The endozoan-based life cycle is something that this is the foundation for our hypotheses as we go forward, right? So this is the small mammal hypothesis. I won't belabor it because Dr. Taylor already talked about this quite a bit. But the basic concept, right, is that sphereal is a dead end because it's not transmissible. So if the sphereal cannot grow out of the lung, then the sphereal represents a dead end, and evolutionarily, that would be being lost from the species if it isn't able to actually get out and replicate and infect the next host. So I'll just close with sort of summary of what we're doing, where we're going in the future. We're doing long-term monitoring of positive sites now. So we've identified a number of positive sites in southern Arizona and in northern Arizona. These sites, as has been mentioned, remain positive for decades. So we're able now to go out monthly, collect soil sample. We've set up air samplers at these sites. We are collecting rodents at these sites. We're doing great epidemiology with Department of Health services. So I think we're finally have the ability to answer these longstanding questions, and we're looking forward to sharing those results, hopefully in the next year. This is work that's being funded by Arizona Board of Regents TRIF funding with Dr. Galgiani, ASU, U of A, others. So it's a big Arizona collaboration to really deeply investigate these sites. So I'll be excited to share those results in the future. So thank you for your attention, and thanks to all the people that do the actual work in my group, and I will take questions at the end. Thank you so much.
Thanks, Bridget.